Hello again, this is Dr. Pflügel. The following presentation will cover the background needed for your next laboratory. This week's lab is the first of a series of 3 exciting laboratories, in which you will analyze your own DNA to determine your mitochondrial DNA haplotype from your maternal lineage. At the end of quarter, you will also build a tree with your DNA and comparable DNA sequences. This lab is the first in a 3-part series in which you will actually collect your own DNA and eventually, quite possibly, learn something very interesting about yourself. The first part in this 3-lab series is to isolate DNA from your cheek cells and prepare your sample for PCR. The samples will then be amplified and sent out for sequencing for use during the last part of this lab series. Since we will be using PCR to amplify your DNA, we will introduce this technique in this week's laboratory, and following presentation will give you some theoretical background on this revolutionary technique. Good primer design is a vital part of developing a PCR protocol, so you will also be asked to practice creating a primer pair in the primer design exercise. The laboratory skills you will meet in this lab include the proper use of micropipetters, Helix extraction of DNA from cheek cells, and using the BLAST online bioinformatics program to check for primer for uniqueness. The concept covered in this lab will include designing a primer pair by hand and by using an online program called Primer 3, which is currently used in research laboratories. Mitochondrial and maternal lineage (PCR) for DNA amplification, and how primers work from 5' to 3' direction. Again, for this week, I would like to ask you to give us some feedback after you have finished the lab. You can do this at any time. However, it would be best if your impressions are fresh when you are giving the feedback. This lab will also continue to enforce standard lab safety. So remember to bring your lab coat and proper lab attire as explained in the lab safety video. Otherwise, you will miss your pre-lab quiz or even a larger part of the lab. This week's lab will focus on a technique called the polymerase chain reaction, or PCR. PCR is probably most famous as the technique that's used at crime scenes or paternity tests, but is tremendously important in day-to-day work in the lab as well. It's contributed to the acceleration of molecular biology and genetics research, and as we will discuss later in this lecture, it's made many other technologies possible. The technique of PCR was first conceptualized in the 1970s, but it didn't really take off until 1983, when Kary Mullis, a scientist working at a biotech firm in the San Francisco Bay Area, developed the modern principle of the technique. For his work, Mullis was awarded the Nobel Prize in Chemistry in 1993. To say the very least, Mullis is quite a character. He's an avid surfer and also very well known to indulge in psychoactive chemicals. In his book, he writes, quote, "Would I have invented PCR if I hadn't taken LSD? I seriously doubt it. I could sit on a DNA molecule and watch the polymers go by. I learned that partly on psychedelic drugs." Unquote. Nevertheless, PCR turned out to be such an important paradigm shift in science that Mullis was awarded the Nobel Prize in Chemistry in 1993. If this man won the Nobel Prize, then so can you. The basic premise of PCR is amplification of nucleic acids, that is, to start with a very small quantity of DNA or RNA, and then build it up to amounts that are usable for other things, like DNA sequencing. In this way, it is useful for applications like crime scenes, where only trace amounts of a sample are available. But an equally useful situation would be, for example, to be able to detect a mutation in a mouse using a very small sample from its skin or its tail. Or, a doctor could use PCR to detect HIV RNA in a sample of blood collected from a patient. For now, we will focus on PCR to amplify DNA. To amplify DNA, 4 things are required. First is a template. This is the DNA of interest that you want to amplify. As we will explain later, when you run PCR on a sample of DNA, you are not amplifying every part of every DNA molecule from every chromosome in the sample. You are only amplifying a relatively short sequence of interest. How do you specify which part of a DNA are you interested in? This is where the second component comes in. PCR also requires a pair of primers, which are typically specifically synthesized. To specify the region of interest to be amplified, the primers should be selected so that they flank the 2 sides of the target region of the interest. In this way, only the sequence between the primers, including the primer sequence itself, will be amplified. We will explain the particulars of this process in a few minutes. Next, you need an enzyme to carry out the amplification. Since you are building DNA molecules, you need a DNA polymerase, which is the polymerase of the polymerase chain reaction. A DNA polymerase is generally an enzyme that uses a single-stranded DNA template to synthesize a complementary strand of DNA based on the base pairing rules. A pairs with T and vice versa, and G pairs with C and vice versa. Finally, since the polymerase builds a new strand of DNA from individual nucleotides, you'll need the 4 nucleotides DATP, DGTP, DCTP, and DTTP as the substrates for the polymerase. With these components, PCR can begin. The first step of the reaction is the denaturation or melting of the DNA sample by applying heat, typically at 94 degrees Celsius. This causes the normally double-stranded DNA to separate into single strands. However, there is a problem. Notice that in this step a very high temperature is needed in order to melt the 2 strands. Like most enzymes in biological systems, DNA polymerases are proteins, and most proteins are denaturated by heat, which disrupts the interactions that maintain the higher order structures. So in theory, new DNA polymerase would have to be added after each melting step, which in practice would make PCR very expensive and labor-intensive. The way to get around this problem was the key improvement to the PCR technique that earned Muller the Nobel Prize. He used the DNA polymerase from the bacterium called Thermus aquaticus, called TAC polymerase from the name of the bacterium. This bacterium was first found in the hot springs in Yellowstone National Park, where temperatures reach about 70 degrees Celsius or about 160 degrees Fahrenheit. This suggested that the bacteria's enzymes were resistant to heat denaturation, and it turned out that the TAC polymerase remains functional even after heated to 94 degrees for brief bursts. This means that the TAC polymerase can be added to the reaction at the beginning and would remain thermostable over the course of the whole reaction. New polymerase no longer needed to be added after each melting step, which dramatically reduced the amount of enzyme and manual labor required. This was the biggest jump that made PCR cheap and efficient enough to be used on a daily basis in the lab, and really drove the development of the applications we'll talk about later in the lecture. Now back to the process of PCR. After the DNA is melted, the temperature is dropped to about 50 to 60 degrees, which allows the primers in the mixture to find and bind to, or anneal, to the regions on the DNA to which they are complementary. Remember that PCR doesn't amplify the entire sample, but only the part in between the primers. Once the primer anneals to the corresponding sequence, it can then be extended by the DNA polymerase into a full-length strand of DNA. At this stage, called the elongation phase, the temperature is raised to about 72 degrees, which is the optimum temperature for Taq polymerase activity. The polymerase will extend the primer into a full-length strand of DNA using the single strand of the sample DNA as a template. The same thing happens on the other strand of the template DNA, where the corresponding primer anneals to the template and is then extended by the polymerase. After enough time has passed for the primer to be elongated, the temperature is raised to 94 degrees and the DNA is melted again. Separating the newly extended primer from the template. At this point, the cycle starts anew. A new primer can bind to these two DNA strands. Let's see how this process goes along for a couple of cycles. As you can see, this results in a preferential increase of the target region of DNA between the two primers. Notice that the small regions of DNA outside the two primers is somewhat amplified as well, but not in the massive exponential way as a region between the primers. If you assume 100% efficiency at each cycle, then the amount of target DNA is doubled at each cycle. So if you run the reaction for a typical 30 cycles, you'll end up with 2 to the 30s, which comes out to be a 1 billion-fold amplification of the target DNA. Of course, the amplification isn't always 100% efficient, meaning that this is a theoretical maximum. Here's an overview of the entire process. Starting with a mixed sample, you use the primers to specify the target DNA to be amplified. At the end of the reaction, you will end up with the target DNA being the dominant sequence in the mixture, meaning that you can then use this DNA for other applications, which we will start talking about in the next part of the lecture. So now you have an amplified sample of your DNA of interest. What can you do with it? In molecular biology, 3 of the most important applications of PCR are genotyping, diagnostics, and gene cloning. The genotyping process involves detecting different alleles or variants of a gene of interest in an individual. Certain alleles could be associated with mutations or transgenes in lab animals, or with inherited diseases in human patients. In these kinds of applications, a very small sample of tissue is collected, typically a small piece of skin or tail in a lab animal, or a cheek swab or blood drawn in humans. The DNA from the sample is isolated, and the specific gene of interest is amplified with PCR. The amplified DNA is then separated by size using electrophoresis in a method similar to the protein gel that we discussed a few weeks ago. Here's an example of a transgenic animal that has had part of a gene for a protein associated with cystic fibrosis deleted, and replaced with a larger selection marker, which is using a knockout animal to model for a human genetic disease. Because of the replacement gene is larger, the DNA length is longer, and therefore runs a shorter distance on the gel. So the top band in this gel represents the diseased allele. Here's a corresponding genotype of a patient with cystic fibrosis compared to a normal patient. In this gel, the lanes labeled N use the primer pair specific to the normal allele, while the M lanes use primers specific to a mutant allele. This genetic test shows that patient 1 is completely healthy, patient 2 has cystic fibrosis, and patient 3 is healthy but is a carrier of the cystic fibrosis allele and can potentially pass it on to his or her children. PCR can also be used to diagnose other types of diseases in addition to inherited genetic diseases. One of the gold standards of HIV testing is based on modification of PCR. HIV is a retrovirus, meaning that its core genetic material is RNA instead of DNA. Since RNA is less stable and harder to handle than DNA, the viral RNA is first converted to an equivalent DNA sequence using the enzyme reverse transcriptase, or RT. This DNA then becomes the template for conventional PCR. To perform the HIV test, a blood sample is taken from the patient. If there are HIV particles in the blood, then the reverse transcriptase will convert the RNA inside the virus into DNA, which will then be amplified by PCR. If the amplified DNA associated with HIV RNA is detected, then the patient may be HIV positive. If not, then it is more likely that the HIV RNA is not present in the patient's blood, and that he or she is HIV negative. Here's an example of what a positive and negative HIV looks like by the RT-PCR method. These 4 lanes here are plasma samples from patients that are known to be HIV positive. This patient has a viral small load small enough to be considered HIV negative, and this patient is completely HIV negative. In addition to detecting DNA sequences, PCR can also be used to add specific sequences to a fragment of DNA. In this modification, the primer pair contains a region of DNA that are complementary to the region of interest, but also additional sequences flanking it that will be added during the elongation step. As a result, the amplified product will still contain the sequence between the primers, but the predominant product will also contain the extra sequence on the far ends of the primer. This could be useful for cloning a gene into a plasmid. For example, you want to insert a gene into a plasmid, but you don't have the right restriction sites flanking your gene. You could use this method to add the required restriction sites on either side of your gene of interest, so you can clone it into your plasmid. Lastly, one of the most famous uses of PCR outside of research and medicine is in forensics, in a process called DNA fingerprinting. Human DNA contains regions of high variability called polymorphic regions, which vary in sequence and length between individuals. 13 polymorphic regions have been identified as markers for this purpose, in addition to a 14th gene that is more strongly expressed in males than in females. Despite them being different among different individuals, the regions flanking these polymorphisms are the same, meaning that they can be amplified by PCR. These 14 markers are then used to match samples between individuals. For example, this can be used to compare DNA samples from a crime scene to different suspects, or DNA samples between a child and a suspected father. In theory, the likelihood of someone having the same fingerprint as you can be as high as 1 in a quintillion. That is about 1 in a million times more than the number of people in the whole world. For this reason, there is a database called CODIS that has been established as a collection of suspects whose DNA fingerprints have been collected over the course of criminal investigations. Of course, it's important to know that because PCR is such a sensitive technique, there is still the possibility of a false positive, meaning that PCR is often not the most critical piece of evidence at a crime scene, as you might always see on the crime shows on TV. In summary, PCR has many more applications that we won't cover here, but it has become one of the most paradigm-shifting techniques for the applications that we have discussed today. For our lab this week, we will not use markers on our nuclear chromosomes, but on the DNA contained in our mitochondria. The DNA in the mitochondria is circular, and what is special about this DNA is that you have gotten it from your mother, and that it doesn't mix with your father's DNA. Therefore, the variation in between individuals is by mutation only. The mitochondrial DNA is a haplotype, which means you have only one copy, in contrast to two copies in your nuclear DNA, where you receive one copy from your father and one copy from your mother. This slide shows 2 people with different types of mitochondrial DNA,
indicating their haplotypes. As you can see, the father's haplotype B and C do not get passed on, and only one type of haplotype remains after only 2 generations. Therefore, your own mitochondrial DNA haplotype comes from your maternal lineage all the way back to a single individual who is called the mitochondrial Eve.
To explain this further, consider this graph here. The spheres signify females and the squares signify males. Here on the top are 10 different individuals with 10 different haplotypes. Whenever parents only have sons, the haplotype doesn't contribute to the lineages in a population and gets segregated out. So after only 3 generations, only haplotype M3 is found in this population. However, remember that most of your DNA comes from the nuclear genome, and even though the 3rd generation only carries mitochondria of haplotype M3, all other genes are a mixture of all of the individuals in the 1st generation.
Since the mitochondrial DNA is a haplotype, each mutation on the mitochondrial DNA can be followed by comparing the mitochondrial DNA in between existing individuals today. For instance, this map shows the relationships of all known mitochondrial DNA of the planet. Each letter indicates a haplogroup, which is a cluster of haplotypes defined by significant sequence changes. The current hypothesis is that the mitochondrial Eve originated in Africa, and then subsequently an individual with haplotype L3 migrated out of Africa and populated the rest of the world. This map shows specific mutations for people with a haplogroup from the various regions in this world. The mitochondrial Eve is postulated to have had a sequence which is called the Recent Sapiens Reference Sequence, or RSRS, which is shown in the lower left corner. This sequence is used as a reference to compare any other haplogroup. To understand the notation of the mutations used in this map, let's focus on the migration out of Africa. This graph shows the mutation distinguishing haplotypes L0, L1, L2, and L3. Each branch introduced a mutation, which then means all individuals belonging to that branch have that mutation. For instance, individuals with haplotype L3 migrated into Europe and then populated the rest of the world. So everyone living today whose recent ancestry is not in Africa carries this mutation and has a C at position 16,278 of their mitochondrial DNA. Whereas individuals who belong to haplogroup L0, L1, and L2 do not have this mutation and have a T at this position in their mitochondrial DNA. Their ancestors were the individuals who stayed in Africa. So by sequencing your mitochondrial DNA, you can find out which branch shown on this map you belong to, or in other words, which haplogroup you belong to. And that is what we will do in lab. This week in lab, you will hypothesize which haplogroup you might belong to from the knowledge of your ancestry, meaning from the history and background of your mother and grandmothers. But remember, even though you might have been born in a certain region of this world, doesn't mean your ancestors were from that region. You have to think about your ancestors from about 10,000 years ago. Once you have a working haplogroup, you will predict the expected mutations for that haplogroup using this simplified map, and then learn how to use bioinformatic tools to visualize and investigate these specific DNA changes. Then, to test your hypothesis about which haplogroup you belong to, you will isolate your DNA from your cheek cells and then amplify and sequence a small region from specifically your mitochondrial DNA.
Shown here is a region on the circular mitochondrial DNA we will be amplifying with PCR. The mitochondrial DNA has a total of 16,589 base pairs, and the control region of the mitochondrial chromosome has two hypervariable regions. We want to look at these hypervariable regions, since it is there that there might be differences in between individuals. We will focus on hypervariable region 1. Here's a detailed map of the hypervariable region 1. The top strand is the reference sequence, and the bottom strand is a complementary strand. The top strand reads 5' to 3' from left to right, whereas the complementary strand, which also reads 5' to 3', is read here from right to left. The green-colored base pairs are conserved, which means they are the same in between all individuals. The yellow regions are base pairs which can be different in between different individuals. In purple is shown the sequence of the forward primer, which has the same sequence as the reference strand on top, and it will bind to the complementary strand on the bottom, and its sequence is read from left to right. In dark blue is shown the reverse primer, which has the same sequence as the complementary strand, and it will bind to a sequence on the reference strand. The sequence of the reverse primer, however, is read from right to left. This distinction is important, since if you wish to order primers for a PCR reaction, you need to indicate its sequence in the 5' to 3' direction. If one wants to amplify a region of DNA which sequence is known with PCR, one has to design a primer pair which will bind to the flanking regions. There are several parameters one needs to keep in mind as listed here for the successful design of a primer pair. Anything can go wrong actually, which simply means the amplification would not work. The specific primer sequence for a specific DNA sequence might differ, however. Knowledge from many experiments indicates that. The length of the primer is important. A size of 20 to 30 bases is optimal. If it's too short, it might not bind specific enough and will bind at multiple places. If it's too long, it might be difficult to anneal to the DNA in terms of entanglement and dynamics for PCR to work. A melting temperature between 55 and 80 degrees Celsius. Guanine and cytosine should make up 50 to 60% of the total base pairs, which helps determine melting temperature. Primers should end at 3' in a G, C, CG or GC, since G and C bind more tightly than A and T, and this will increase primer specificity. Long runs, 4 or more of the same base in a row, should be avoided, since this can cause the primer to bind in the wrong place. For example, let's look at the reverse primer we will be using for the DNA amplification in lab. The 5' end of the reverse primer is binding to a region on the DNA which is a variable site, here shown in yellow. How would this affect the PCR? A mismatch at the 5' end of the primer as shown here might affect the melting temperature of the primer, which means it might dissociate at a lower temperature. In this case, the DNA polymerase can still bind and extend the primer in the 3' direction. However, a mismatch on the 3' end of the primer might render the PCR unsuccessful. The third case, a mismatch in the center of the primer, again might change the melting temperature and or render the PCR reaction inefficient. In the early days, primers were empirically designed by hand. Nowadays, however, one can use programs to find the best design for a PCR reaction if the target sequence is known. One of these programs used in the laboratory is Primer 3. In this week's lab, you will use Primer 3 to propose some designs, which you will use then to analyze in terms of usability. Here's an example Primer 3 result page for the primers we will actually be using to amplify your mitochondrial DNA of hypervariable region 1. This program takes as input the sequence of the target DNA and optional the primer sequences. Then the program will output where the primer sequences will bind on the target DNA, how long the primers are, their melting temperature, GC percent, and two more parameters indicated here as NE and 3'. Furthermore, it will indicate the size of the PCR product, and again, two more parameters indicated here as pair complement and pair 3'. Before I explain these extra parameters, one thing to look for here is the melting temperature of the primer pair. Since the primers work in tandem, it follows that their melting temperature should be similar, which they are. They have a difference of 1.3 degree in their melting temperature here. Now, the parameter NE indicated in the table will give the number of bases which a primer will fold back onto itself and pair up, as shown here. This number should be small for a good primer. 3' indicates the number of base pairs a primer will bind to another primer of the same type. Again, this number should be small for a good primer. The pair 3' complement indicates the same for a forward primer binding to the reverse primer. Again, this number should be small for a good primer. When you investigate your own primer design, you want to ask some questions, shown here. Is your primer design unique? Does it bind in regions that are highly variable? Does it bind to itself? How good do you think your primer is overall? All this information you will find on the lab website under this address, if you want to look at the information posted beforehand. I will end this lecture again with a reminder to wear your appropriate PPE in lab next week. For this lab, you need to wear goggles, gloves, and a lab coat, besides the standard lab attire to fulfill lab safety requirements. Here's a summary safety slide you can study before coming to lab. Thank you for your attention and enjoy this week's lab.